Welcome children. In this video I will be teaching phytohormones, and the strategy I am opting to teach is using mnemonics. You all are aware with the different strategies like auditory or visuals, reading, writing or kinesthetics. I am opting the best strategy, that is mnemonics, which will aid your memory. It improves your memory. So, there is no other option rather than mnemonics. Maybe knowingly or unknowingly you must have executed mnemonics in 10th standard for social studies, to remember terms, to remember hierarchy, etc. It's just either you learn it as a phrase or a sentence. For example, a mnemonic, commonly used mnemonic in maths is BODMAS. B stands for bracket, D stands for division, M stands for multiplication, A addition, subtraction. So, it's the order of operations. Same way to learn the order of the planets, we learn a mnemonic to get the correct order. First Mercury, then Venus, then Earth, Mars. To get that order, we remember a mnemonic, which usually teachers tell you all. A sentence: my very excited mother just served us noodles. So, you will get the correct order of the 9 planets. So, and also I have implemented mnemonics during the course of our study. But I did not introduce this term because only partly it was introduced. But today's class is exclusively tricks and tips to remember using mnemonics. Okay. For example, the RBC loaded with oxygen and carbon dioxide is a mnemonic. I gave it a symbolic mnemonic. In a circle, I made that carbon dioxide enter, and then it got split, and then it combines, formed reactions, so that you will never forget. So, mnemonic is a psychological aid which will aid or improve our memory. So, this whole instruction goes with mnemonics. And we know phytohormones, there are 5 categories of phytohormones: auxins, gibberellins, cytokinins, ethylene and abscisic acid. So, all 5 I will be executing mnemonics. Let us start. So, first I am going to deal with the phytohormone auxins. It was first discovered or isolated by F.W. Wendt. And for this I am taking as a mnemonic, for you to remember, I am taking the entire part of the plant, a basic plant's body, which has roots, stem, leaves, flowers, fruits, buds. So, with every part we have a characteristic, or the particular phytohormone called auxins have a role to play. So, the mnemonic I am going to use is a basic plant with all the parts. So, the features or the different functions auxins controls. First, starting from the base of the plant, the first function I have taking the plant as a mnemonic for you to memorize. I am starting from down to top. It initiates, auxins initiates rooting in stem cuttings. So, we know some plants are propagated via the stem, and there when we add auxins, rooting is propagated or rooting is initiated. So, that is aided by auxins, rooting in stem cuttings. So, I have dealt with roots. Next, I have gone to the shoot part. Okay. We know there are 2 types of buds in plants. Okay, on the stem there are 2 buds, one bud at the terminal end, and that bud is called the terminal bud, or it is also called apical bud, and the bud found at the axil of the leaf petiole and the stem. See, at the angle there is known as the axillary bud or lateral bud. So, we identified 2 buds, one at the tip of the stem and one at the axil of the leaf and the stem, angle of the leaf and the stem. Now, here the auxins promote apical dominance. The meaning of apical dominance is the apical bud is dominating the lateral bud, which means the apical bud takes over and allows the growth of the plant vertically. It grows taller than broader because it suppresses the activity of the lateral buds. So, here and there I am drawing lateral buds. This is suppressed. The functioning or the activity of the lateral bud is suppressed by the apical bud. So, such types of plants will grow taller than broader, and they have a cone shaped canopy. Okay. Next, third, it induces parthenocopy in tomatoes. So let's identify in this plant a tomato. Okay, this is tomato. And we know, and we'll be studying in 12th sem what is parthenocopy. It is fruit formation without fertilization. So, auxins promote parthenocopy in tomato. So, we dealt with the fruit part. So, I am ticking the fruit part. Next, fourth point, auxin promotes flowering in pineapple. Look at the plant I selected. I selected a plant which bears yellow flowers, so that you will remember which fruit it induces flowering. It is a pineapple. Yes, of course, there are some other fruits which are yellow in color, but here you should remember it's pineapple. It initiates or promotes flowering in pineapple. So, let this be the pineapple flower. Because this is a mnemonic, I will take a basic plant. Next, prevents early leaf and fruit fall. So, regarding the leaf and the other fruits, it prevents early falling. So, the leaves are intact and remains on the tree or plant for a longer time because of auxin. So, we have covered all the parts. If you verify the mnemonic, we have covered all the parts. We have covered the root. From the root perspective, what did I say? Auxins initiates rooting in stem cuttings. With regard to the stem, what did I say? It promotes apical dominance. Then, with regard to fruit, what did I say? It promotes parthenocopy in tomato. Regarding flowers, it promotes flowering in pineapple. And then what is left? The part of the plant that is left behind is the leaves. Leaves and other flowers and buds. It prevents. So, regarding this, it prevents early falling, premature falling. So, all the parts of the plant are touched. So, this helps in getting all the role of auxins in plant, because it's including all the parts, the root, stem, leaves, flowers, fruits. But it's specific to a particular flower or fruit. For example, fruit, it's not all fruits, parthenocopy in tomato. In flower, it is flower formation in pineapple. Okay, so that is the mnemonic used for auxins. So, you will never forget what comes to your mind. Don't go for rote memory. You go by this mnemonic. Think of parts of a plant starting from the root, then you take the stem, then the leaves, flowers and fruits. You get all the 5 functions of auxins. Next, moving on to the different types of auxins. They are IAA, IBA, NAA and 2,4-D. IAA stands for indole acetic acid. IBA stands for indole butyric acid. NAA stands for naphthalene acetic acid. 2,4-dichlorophenoxyacetic acid is 2,4-D. Of these, the first two, IAA and IBA, are natural auxins, and the remaining two, NAA and 2,4-D, are synthetic or artificial made auxins. Clear? So, some examples: IAA, IBA, NAA and 2,4-D. Of these, our gardeners, our gardeners, when they make a lawn, it is spectacular. It's very beautiful. It's just like a green carpet, and you'll be wondering, it looks like an artificial green carpet. But the reason is the trick behind this is that in the fertilizers, in the manure they use, they mix 2,4-D. The advantage of 2,4-D is it prevents dicot weeds from growing. So, it is best used to make weed-free lawns, and usually the weeds are dicot. So, we can get a lawn free of these dicotless weeds. So, we have only exclusively the particular grass, whether it's a buffalo grass or Singapore grass or the needle grass. It looks clear like a green carpet because 2,4-D is mixed in the manure and applied, and 2,4-D is a weedicide. It only targets the dicots, the dicot weeds, not the monocot or the grass. Clear? So, that is auxins. Once again I am reiterating, go with the mnemonic. the basic parts of a plant with stem, roots, leaves, flowers and fruits. Now moving on to the next, phytohormone. The next phytohormone is ethylene. It was discovered by cousins, and here I wish to draw the mnemonic. Okay, we know that I am starting the life of a plant from the bud. Usually we start the life cycle of a plant from the seed. I am just starting from the bud. See, the bud, you know what is bud? The petals all close together. Okay, the bud is closed, and what happens the next stage of the bud? It blooms into the flower, and maybe this flower, whatever, bisexual or unisexual, it is fertilized by pollination. And what is the result of fertilization? Fruiting. Fruit, and within the fruit seeds are formed. Now the seeds fall on the ground or on the substratum and it germinates. We know usually when we start the life cycle of a plant, we start with the seeds falling on the ground, or we sow the seeds and then it germinates into the root system and shoot system. But I started, the mnemonic here is with first bud. From the bud what arises? The flower arises. From the flower, after fertilization, it gives rise to the fruit, and within the fruit there are the seeds, and when the seeds fall off from the plant, they germinate. They germinate, how is it? Root is formed as well as shoot is formed. Now at every aspect, at every level, there is a role of ethylene. So let's start. At bud, you just put this indicator. I put a cross against bud and the other end of the flowchart seeds. The meaning of that indicator, let it be breaks. So here it indicates breaking bud dormancy, and here it indicates breaking seed dormancy. So what is dormancy? Dormancy is sleep. Actually in French it is called just sleep. In plants, what does it mean? It's an inactive state, temporary inactive state. So sometimes the buds go dormant, inactive. It's not dead, it's inactive. But the role of ethylene, if ethylene is applied on such plants where the buds are dormant, it breaks bud dormancy. Meaning of breaking bud dormancy means it initiates blooming of the buds. So the actual effect, it helps in the blooming of the bud. Similarly, seed dormancy. I told you again, it's breaking seed dormancy. So what is it actually doing? It is hastening or promoting seed germination. So opposite of dormancy is, in case of seed, it is germination, seed germination. In case of bud, opposite of bud dormancy means bud blooming. So I am going to start. The first role is breaks bud dormancy. You can interpret in the other way, in the positive statement, that is, it initiates blooming of buds into flowers. Next, I am going to this level, flower. It initiates flowering in mango. So if we apply this ethylene on mango plants or trees, what happens? It initiates flowering in mangoes. Next, moving on to fruit. It hastens fruiting in apples and tomatoes. So it makes that fruiting process faster in apples and tomatoes. In fact, in all it's applied in all fruits to make this process of fruiting faster. Next, as I've already indicated, breaks seed dormancy, or you can talk in terms of positive. That is in a negative statement. Here it initiates seed germination. Now I have reached the fourth role, it's over. Now next we are having roots to be dealt with. It promotes root growth and root hair formation. So what is the advantage when the root grows and the root hair flourishes and extends and it becomes elaborate? The advantage is for the plants. It increases the surface area of absorption, because it is only through the roots that the plants absorb nutrients. So when the root grows and its lateral branches increases in number, what happens? The surface area increases. So it's advantageous to the plant. And lastly, about the shoot system. Here I wish to remind you about the paddy cultivation. We have seen, and we especially Kerala, we are blessed with a lot of farms where we are involved in paddy cultivation, and you know what type of soil is used, the clayey soil. And why such clayey soil is used? Because paddy requires stagnant waters. Okay, in the Kuttanad belt and all, you can see paddy cultivation, and it is in standing waters. There are two types of paddy crops. One that requires less water, so they are called shallow water rice crops. And the others that require more water, stagnant water, they are called deep water rice crops. So if we have a substratum with less water, then which type of crop we have to sow, which type of paddy we have to sow in that particular substratum? We have to definitely sow the rice which requires shallow water, that is the shallow water rice crops. So I'm talking about the two rice crops, shallow water rice crop. And suppose the substratum has a lot of water, standing water, we go for deep water rice crops. So there are two types of rice crops. Now in the case of deep water rice crops, there's chance of decay, because that water should be standing. If the water recedes, what will happen? Or if the water becomes more overlogged, what will happen? The plant will get fully submerged and there's chance of decay. So we should be very sure, make sure that the shoot system is above the level of the water. In that case, we apply ethylene. So it's always advisable to apply ethylene in standing waters or in paddy cultivation, especially in the case of deep water paddy crops. Even in shallow water paddy crops it's good, but the water is very shallow and maybe those areas can become arid as early as possible. That's why the shallow water. But deep water rice crops just high harvest, and to see that the plant does not get submerged, we have to apply ethylene, because the role of ethylene is, suppose I draw this rice, we know it's a monocot. This is one leaf, this is the next leaf and the other leaf. And remember, once again I am redrawing the leaf, it has a petiole and then the leaf. Petiole and then the leaf. And then there is a common stem. So all with the petiole. Now it is in the water, so I am putting blue. Now if the water level rises, see it raised, the plant will get submerged. This should not happen. So to lift, we can't lift the plant. So in that case, what is done is ethylene is applied. When ethylene is applied, what happens is, I am drawing the plant this side. The stem internodes elongates. You know what are internodes? In the stem there are the regions called the nodes, and the gap between adjacent nodes are called internodes. They elongate, so it became tall. Not only that, the petioles also elongate. They elongate. And then the leaf or the shoot system arises. So there's no chance of the plant to be submerged. It's well above the water body. The leaf is well above the water body, or the shoot is well above the water. But in this case, you can see it is getting fully submerged. Why it is getting fully submerged? The stem is short. See, the stem is short, the petiole is also short. So what we have to do, just apply ethylene. When we apply ethylene, what happens to the petiole? See how, look at the petiole, it was so short, it became very lengthy. Then also look at the stem, it was short and it became lengthy. So long it became. So that we can make sure that the leaf system arises or remains above the level of the water and prevents decaying of the plant. So that's the point. promotes rapid elongation of internodes and petiole of rice, and specify which rice. It is the deep water rice varieties. Clear? So, once again brushing through the functions of ethylene. 1. It breaks bud dormancy. 2. It initiates flowering in mangoes. 3. Hastens fruiting in apples and tomatoes. 4. Breaks seed dormancy. 5. Promotes rapid elongation of internode and petiole of rice of deep waters. And 6. Promotes root growth and root hair formation, therefore increases the surface area of absorption. So, with this mnemonic, so the mnemonic I used is this, the life cycle of the plant starting with the bud. So, I will just highlight the mnemonic before going to the next page. So, I gave the life cycle starting with the bud blooms into the flower gets fertilized and becomes the fruit, and in the fruit there is the seed, and the seed germinates to form the shoot system and the root system. Clear? This is the best mnemonic where you can get all the functions of ethylene. And remember, ethylene is a gaseous hormone, or it is a volatile hormone. Next, moving on to gibberellins. Gibberellins was discovered by E. Kurosawa. And the functions of gibberellins, what should come to your mind is a list of these fruits: apple, grapes, sugarcane, and one vegetable, that is cabbage. Now, the role. When you go to the market, which type of apple do you purchase? Naturally, we go for the red apple. We get attracted. It's very appealing to the customers. Other than that, the shape which we opt. Definitely, we all will answer that we go for the round shape, globular shape. But actual shape that is appealing to the customer is the oblong or the elongated shape. How is this elongated shape of the apple made? That is by applying gibberellins to the plants before fruiting. So, to elongate, so the first role, to elongate the apple and improve its shape, first function. So, we get elongated apples which are more in demand than the round apples. Second, it is applied in the vineyard so that the grape stalks increases in length. And what is the advantage of such grape stalks? See, I am highlighting the grape stalk here. It's lengthy. Look at this, it's also very lengthy, and here also we have a lengthy grape stalk. If it was short, it would have been very difficult at the time of harvesting. It will be very difficult in the time of to allow it to creep on the climbers. And also it is now used for ornamental purpose, like fresh flower arrangement. We have fresh fruit arrangement where stages are decorated with grapes, and the grapes that are preferred are the grapes with long stalks. So, this is in demand to increase the length of grape stalks. Then third, to increase the length of the stem of sugarcane. Look at sugarcane. The length and width of the sugarcane increases, definitely the yield of sugar increases. That is the purpose. When we apply gibberellins to sugarcane, what happens is the stunted sugarcane also become elongated, and therefore the yield of sugar becomes many times high. Then one with respect to the vegetables which are in rosette-like habitat. Rosette-like habitat, have you seen cabbage? It is a leaf, it's a leafy vegetable, and you can see if I draw the internodes, basic internodes or nodes of a plant, these are the nodes, and the region between two nodes are called the internodes, and we can see from one node the leaf arises. The nodes give rise to the leaf. Now, in cabbage we can see this gap is not there. You can see the leaves are overlapping one another. Okay, without any gaps between the internodes. So, now the use of gibberellin is to promote bolting. What is bolting? Internode elongation. You must have seen the Chinese cabbage. Chinese cabbage is like this way. There is the internode between the leaves, very prominent. So they look very fluffy. That is more appealing to the customers. They prefer the Chinese cabbage more than the Indian cabbage. So now what we have done is we have modified the Indian cabbage. We have applied a lot of gibberellins before it could form this rosette fashion. And now we can see gaps between the leaves. So this is one leaf. The next leaf that is overlapping will have a gap. Then next leaf, next leaf. That way it becomes very fluffy in appearance. Not tightly packed like the traditional cabbage, which we still use. Okay, overlapping of the leaves tightly. Okay, and we know center there's the internode which is not used for, which is not edible. We just get rid of that. So you are familiar of cabbage. So to promote bolting. So what is bolting? Internode elongation in plants like cabbage, beet, etc., which has rosette-like habitat. I hope now you understand rosette-like habitat. The leaves are not arranged like say laterally. This is lateral arrangement, but you can see it like a whirl. It is arranged and one over the other, piled up into a globular structure. And lastly, which I didn't incorporate into the slide, but it's a very, very important point. I am writing the fifth point on that dark part of the slide in white. The fifth importance of gibberellin, it delays senescence. So that the fruit remains on the tree. So as to extend the market period. For example, a very good example is tomatoes. When you purchase tomatoes from the market, definitely we go for ripe tomatoes. No one will purchase raw tomatoes if for a particular purpose of making a particular curry, but usually what is purchased is tomatoes, and we don't buy tomatoes in bulk. We only select a few tomatoes, they ripen and we use it within a day or two. It will not go beyond. The problem of tomatoes within the market also, they stock only less, because we cannot, unlike cereals and pulses, tomatoes gets rotten and spoiled easily and we cannot stock it. Even the market do not have place to stock tomatoes. They get easily oozed out and it's not fit to be used. When we also have noticed in our house, more than 4 days tomato will get spoiled. So in that case, there's lots of problem regarding stocking and selling of tomatoes. So what is done? Gibberellins are used. It's applied on the plant. So the tomatoes which are going to ripe are stuck. They are static. They do not ripe. It delays their maturity. Their maturity is on hold. They are not allowed to mature. That is called senescence, delays senescence, and it's left on the tree for until it's required. So when the market is almost, if there's clearance of the tomatoes, then we stop applying gibberellins and it naturally ripens, and then we go for a harvest of tomatoes. So this is always met with tomato cultivation, this problem, okay, because tomato is a perishable good and very fast it's being perished because of this property. It gets spoiled very easily, it oozes out and the juice comes out and spoils the tomatoes in the immediate surroundings. So the best option is to apply gibberellins. There's no silos, there's no granaries, there's no stocking for tomatoes. So what we do is we apply spray gibberellins on these plants or trees, and therefore the plant delays senescence and therefore the fruit remains on the tree, and therefore it extends its market period, its longest shelf life. And when in the market it's time for a new harvest, that time gibberellins is stopped applied and this naturally ripens and we can harvest. Okay, so last point, don't forget to incorporate delays senescence. Therefore fruits remain on the trees for a longer period to extend the market period. That's very important. So once again, the functions of gibberellins: elongate the shape and thus improve the shape of apples, specifically for apples. Second, it increases the length of grape stalks. And I told you the purpose of grapes. Grapes, other than being eaten, it is used for decoration, and such stalk grapes are advantageous. If they have longer stalk, it's easier and preferred. Then it increases the length of the stem of sugarcane, therefore the yield increases. And lastly, promotes bolting in rosette-like vegetables like cabbage, beet, etc. That is internode elongation. Therefore, we get fluffy cabbage like the Chinese cabbage. And last of all, delay senescence. So, the fruit remains on the tree for a longer period until required in the market. Next. Now, before going to gibberellins, remember gibberellins are named or denoted by alphabet G1, GA1, GA2, GA3 and so on. Among these gibberellic acid, GA3 is commonly used for malting drinks in the brewer industry. So, in brewing industry, distillation, in brewing industry, this is used GA to speed up the malting process. Okay, we know where malting is used. Okay. Next, cytokinins. Here I have opted for a mnemonic. One is to break it from the word itself, and the second is because the cytokinins is an antagonistic hormone to auxins. So, the mnemonics I selected here is from the very name cyto, break cytokinins, cytokinins. What is the meaning of cytokinins? Cyto stands for cell and kinin stands for division. So, cytokinins, the first and foremost function is it promotes cell division. Next, as I told you, the second and third point will go in context to auxins because cytokinins are opposite or antagonistic to auxins. I gave the symbol opposite sign, which means antagonistic. If it was having the sign equal to means it's synergistic. Now here auxins and cytokinins are antagonistic, opposite in function. So with that aspect in mind, let's see the second feature. Promotes, so recollect, auxins promote rooting. So here opposite of rooting, don't say shooting, the better word shoot growth. Promotes shoot growth in callus. Callus means in tissue culture. And the third function, which is also antagonistic to auxins, we know auxins promote apical dominance, and as a result of apical dominance we see the tree canopy like this. This is because of apical dominance. This is due to auxins, and I told you cytokinins is opposite. So it promotes lateral dominance. So look at the canopy of the tree, it's like an umbrella because the lateral bud is more dominating and suppresses the activity of the apical bud, whereas here the apical bud is dominating and suppresses the activity of the lateral bud. So this lateral dominance is promoted by cytokinins. So once again, the three important functions of cytokinins, one you will get it from the name by splitting the name. Cytokinins means cell division. So it promotes cell division in callus or in the plant itself, wherever it has to grow, it promotes cell division. Second, it promotes shoot growth in calluses. For example, that goes in context to auxins. Auxins promote rooting. Here it promotes stem growth, especially in the case of tissue culture. You know what is tissue culture? We will be learning in 12th standard. A small piece of the tissue is taken from the plant and put in a culture medium, and then it grows and becomes a mass of cells called the callus. Here I am having the picture of a callus. Okay, just a bulk or a mass of cells which is not differentiated. Now we have to differentiate it. So what we have to do, we have to apply auxins and cytokinins. Both should be applied in a specific ratio. Auxins and cytokinins should be applied. So when auxins are applied, it promotes rooting. Okay, and when cytokinins are applied, it promotes stem growth or the shoot formation. Okay, so both are mandatory, should be supplemented for tissue culture. Okay. So finally we have thousands of plantlets formed with the application of both auxins and cytokinins. So these two hormones are very, very important in tissue culture. Next, The third aspect is promotes lateral dominance. Again, in context to auxins, it is opposite to auxins. Auxins promote apical dominance. This one promotes lateral dominance. Now, cytokinins was discovered by Scooge and Miller. That also you should remember. Who discovered, two scientists discovered cytokinins, namely Scooge and Miller. So, the mnemonics I have used here, remember, it is antagonistic to auxins, and you are taking the two features which is opposite to auxins. In auxins promotes rooting, here it promotes shoot growth. Auxins promote apical dominance, here it promotes lateral dominance. So, you got 2 points from antagonistic nature to auxins, and the third or the next point will be from the very name, split the name, cytokinins, promotes cell division. So, this is clear. Next, moving on to abscisic acid, the same mnemonic I am using here. Split as abscisic acid, not split the meaning of abscisic acid. We need not split. Abscisic acid is a meaning, is the English name itself. Abscission. What's the meaning of abscission? Abscission means falling. So, the meaning from the very name itself you will get one function. Abscisic acid promotes abscission of leaves, flowers, fruits, etc. So, I have showed you of a dense forest with foliage. Okay. So, not leaf, flowers, fruits, everything falls because of abscisic acid. So, it is a negative, it is an inhibitor. Next, the next mnemonic I have used. First mnemonic is from the meaning of the hormone, which means abscission. The second mnemonic, it is nicknamed. The nickname, it is called stress hormone. Why it is called stress hormone? So, I am giving here abscisic acid or stress hormone. Why it is called stress hormone? So, the stress that plant face is water stress. So, I have taken out a pull out of a plant which shows water stress. This plant A shows abundance of water. See, and because water is abundant, you can see it does not hinder photosynthesis. It grows, and look at the leaf lamina, broad and healthy plant. Okay, but what happens if the plant faces water shortage? That is the stress here. What will happen is, the first thing, the plant has an inbuilt mechanism to close the stomata. So, here you can see the stomata is wide open, but here what happens when this water shortage in the substratum, the stomata closes. When the stomata closes, what happens? Will carbon dioxide be taken? No. Will hence photosynthesis occur? No. So, what will happen to the leaves? It all gets wilted. So, this is because of abscisic acid. The closure is brought about by abscisic acid. Abscisic acid is responsible for tiding over this condition, to cope this condition. Just imagine if abscisic acid was not there. The stomata would have been opened, and when the stomata is opened, what will happen? The water, whatever water was there, will be lost by transpiration. The closure of the stomata prevents water loss by transpiration. So, abscisic acid there helps in storing or saving the water. How it helps in saving the water? By enabling the closure of stomata. So, the stomata all closed. Whether photosynthesis does not take place or takes place, it is not a matter. Definitely photosynthesis will not take place because the stomata shuts. But it is a strategy to tide over, to cope up with the scarcity of water. So, in short, we can say it causes closure of stomata under water stress, so as to prevent water loss by transpiration. So, definitely it is a stress hormone because it enables the plant to survive the stress. Then on return of the favorable season, abscisic acid is not released. Then we can see the stomata opens and the plant recoups. Clear? So, it helps the plant in such scarce condition, adverse condition like water stress. It closes the stomata. Next, suppose it's a plant which bears fruits and the fruits ripens and seeds fall on the substratum. So I just include a seed here, and this seed fell on this substratum, not on this substratum where there is abundant water. It fell on the substratum where there is water shortage. Now what will happen? Will the seed germinate? If at all the seed germinates, it will be an unhealthy seed, very unhealthy. Unhealthy seed will be formed. So what does the abscisic acid does? It inhibits seed germination, or it promotes dormancy of seeds. So the advantage of being dormant is to cope or to tide over it enables the seed to tide over that unfavorable condition. If at all it germinates into an unhealthy plant which will not survive. So what happens? It becomes dormant. Actually it is alive but inactive. It becomes active or viable on return of favorable season. When the water is plenty, then it will germinate into a healthy seedling rather than doing it early. So that is a strategy to again tide over stress, again the water stress. So causes seed dormancy under water stress. So therefore, do you agree that abscisic acid is a stress hormone? Yes. So once again, abscisic acid, the first function you get of abscisic acid is from the meaning of abscisic acid. Abscisic acid means abscission. So it promotes abscission of leaves, flowers, fruits, etc. Next, it is also called stress hormone. So we have to justify why it is called a stress hormone. And remember, the stress here, it is water stress. So how it acts as a stress hormone? When there is water scarcity, all the stomatas are closed because of abscisic acid. And why it is stored? It's closed to save the loss of water, to not allow the water loss by transpiration. Therefore, the water is saved. Just imagine the stomatas open, lot of water will be lost and the plant will die fast. So this is an adaptation where the plant survives and overcome that unfavorable condition by closing of the stomata, which is brought about by abscisic acid. Similarly, if a seed falls on the substratum, it happens to be very dry, that seed is not going to germinate. At the same time, the seed is alive but not active. So that condition is called dormancy. It's brought about by abscisic acid. So this dormancy is produced or created in that seed by abscisic acid. A lot of abscisic acid is produced, and therefore that seed will not germinate. And when there's abundant water, slowly abscisic acid will stop being released and the seed will germinate. So therefore it's also called a stress hormone. Now abscisic acid was in 1960s was discovered by 3 independent scientists, and these 3 scientists gave this particular hormone. They named it by different names. One named it as inhibitor, another person named it as abscission, another person named it as a negative hormone. So 3 independent scientists gave different names, and finally they identified that the property was the same, and finally they decided to give the name abscisic acid from the very function it does, abscission. So of all the hormones we have learned so far, can you say which we can categorize it into 2 categories, promoters and inhibitors? So which can be the promoter? If you take a review, which can be promoters? Auxins, yes. Cytokinins, yes. And gibberellins. So of the 5 hormones, 3 are promoters: auxins, gibberellins and cytokinins. The remaining 2, abscisic acid is an inhibitor. The one which is shown, it's an inhibitor, inhibiting photosynthesis, inhibiting seed germination, making the leaves and all to fall. So it's inhibiting, negative impact on the plant. Though it is a positive for the betterment of the plant, the role is inhibition. So it's a negative. So it serves as an inhibitor. Ethylene, in that case we can't place ethylene into any of the 2 categories, whether it's a promoter or inhibitor. It is between these 2 categories. The reason is that, I'll just go to the slide of ethylene. Ethylene is put under promoters as well as regulators. Promoters, when we see in the case of initiate flowering, hastens fruiting, breaks seed dormancy, breaks bud dormancy, promotes rapid promotes. Everything is promoting. So it should be positive. But why do we say ethylene is also negative? Because now I have to include 2 down arrows. That is, it initiates even abscission of flowers and fruits. So in your mind map or in your mnemonic, please include the down arrow. Down arrow indicates falling. So ethylene will be complete with one more function, brings about 7th function, brings about abscission. I purposely brought this because we had to place ethylene either as a promoter or as an inhibitor. With all these functions listed on the slide, it serves like a promoter. Then why does this confusion or dispute? Ethylene is also an inhibitor. This is the reason. It promotes, it also plays an important role in abscission of flowers and abscission of fruits. And remember the 2 F's here. Okay, not least flowers and fruits, it brings about abscission. So in that context, it is both negative as well as having positive role. Clear? So once again, I am wrapping up by saying the 5 hormones, phytohormones or plant growth regulators. They are also known as PGRs. They broadly 5 types: auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Of these, the first 3, auxins, gibberellins, and cytokinins, are surely promoters. Abscisic acid surely is an inhibitor, and ethylene's position is either it can be put as a regulator, as a promoter, as well as an inhibitor. Promoter with respect to all what was discussed. An inhibitor, what was included now purposefully at the end, that it also, like abscisic acid, brings about abscission of flowers and fruits. So don't forget to incorporate that and get all the functions of ethylene, which is a volatile hormone. I hope you have understood with the help of mnemonics. Thank you.